Hello student, good afternoon. Today we will start the general characterization and classification up to class level of Protozoa. The name Protozoa comes from the Goldfuss in 18, who however used the name simply for the lower groups of the zoophyta. Includes protozoans, sponges, coelentrates, rotifers, and bryozoans. The Horn-Siebold 1845, recognizing the unicellular nature of the protozoa. Protozoa are generally microscopic in size. They are found in freshwater, salt water, and damp soil. Some protozoans are parasitic, symbiotic, commensals. They are called acellular or non-cellular because their bodies are not differentiated into cells. Protozoa forms a heterogeneous groups. Protozoa forms the heterogeneous groups. The members display an extreme diversity of structure, different type of symmetry, and adaptations to various environments. Many protozoa are extremely complicated in structures. Same protozoa form two colonies. A protozoa might be defined as an organism which is made up of mass of protoplasms not divided into cells and which carries on all the vital activities of life such as locomotion, feeding, digestion, egestion, respiration, excretion, and reproduction, etc. Protozoans are simple animals. Protozoans are simple animals. For many have complicated structures and are physiological complex. In as much as protozoa are not made up of cells, they represent that is called the protoplasmic level of organization. Some protozoa are quite close to the plants and may be considerable as connecting link between animals and plants. The number and variety of protozoa is very great. About 50,000 species have been named, although over 20,000 of them are fossil forms. The immense number of protozoa have traditionally been separated by their means of locomotion into four classes: Sarcodina with pseudopodia, Maltigophora with flagella, Ciliata with cilia, and Sporozoa with no organelles for locomotion. Recently, the Committee on Taxonomy and Taxonomic Problems of the Society of Protozoologists published a revised classification of the protozoa. Hangingberg et al. 1964. According to Hangingberg et al., protozoa have been divided into four subphyla: Sarcomastigophora, Sporozoa, Nidospora, and Ciliospora. For example, Euglena, Trypanosoma, Amoeba, Plasmodium, Paramecium, Entamoeba, etc. Nutrition may be holozoic, holophytic, saprozoic, or parasitic. Intracellular digestion takes place inside the food vacuoles. Body oval, spherical, elongated, or flattened. Respiration occurs by diffusion through general body surface, excretion through by permanent pore. The cytophiles, contractile vacuoles, performs osmoregulation in freshwater form and also help in removing excretory products. Reproduction asexual or sexual. Asexual occurs by binary multiple fission, budding or sporulation, and sexual is performed by gametes formation or conjugation. Life cycle often exhibits alternation of generation is includes asexual and sexual phases. Classification up to class level. The classification of Protozoa divided into 4 subphylum according to the Königsberg et al. 1964. Sarcomastigophora is the order. Sarcomastigophoria. Mastigophora superclass. Phytomastigophoria class. Zoomastigophora class. Opalinata superclass. Sarcodina superclass. Rhizopodia, Pyroplasmia, Actinopodia, Sporozoa. Tylosporia class. Toxoplasmia class. Haplosporia class. Nidospora, Myxosporidia, Microsporidia. Ciliophora ciliata. Ciliophora ciliata. Sarcomastigophora. Organelles of locomotion are pseudopodia or flagella. Pseudopodia or flagella. Nucleus is of single type, monomorphic. There is no spore formation. Syngamy occurs in reproduction. Mastigophoria. They are commonly called flagellates. Organelles of locomotion in adults are flagella. Body is covered by pellicle. Binary fission is longitudinal. They are mostly free living, though some are parasitic. Example, Mastigomia. They generally possess chromatophores. There are usually only one or two flagella. The nucleus is vesicular. They have no chromatophores. There are one to many flagella. Often there is an undulating membrane. Most of them are parasitic. Opelina. They have numerous cilia like organelles in oblique rows over the entire body surface. There is no cytostome. Two or more monomorphic nuclei are present. Binary fission is Kinetal, there is syngamy with flagellated anisogametes, all are parasitic. Sarcodina, their organelles of locomotion are pseudopodia. The amoeboid form is pseudominant. Some have a hard shell. They generally do not form spores. Formation of gametes and flagellates. Young ones are common. Their organelles of locomotion are logopodia or philopodia, but never oxopodia. They are generally creeping forms, small round rod-shaped or amoeboid parasites in vertebrates, red blood cells, example Babesia. Their organelles of locomotion are delicates and radios oxopodia. They are primarily sessile or floating forms. Test is present or absent. Gametes are usually flagellates. Reproduction is both sexual and asexual. Sporozoa. The adult has no external organelles of locomotion. They are all parasitic and incapable of active life outside the host. Cilia or flagella may be present in gametes. Syngamy takes place, after which many spores are formed. The spores are simple and contain one to many sporozoites. Sporozoites are the infective phase. Nucleus is of the single type. Single type. Okay, this is the outline classification of protozoa. Outline classification. General characters and outline classification of protozoa. This is the remaining part to be continued in next period. Okay, thank you.